Now we're going to hear from Stephanie Bivona, who is a board-certified genetic counselor at the University of Miami Miller School of Medicine. She graduated with her BA in general biology from Washington and Jefferson College in 2015 and her MS in human genetics and genetic counseling from Stanford University in 2017. Stephanie specializes in neurogenetics and is a part of the Pediatric and Adult Muscular Dystrophy Association Clinic at the University of Miami. Stephanie also works with neurological conditions, included on Florida's newborn screening program and the Huntington Disease Center of Excellence at the University of Miami. She is the clinical site coordinator of the NIH-funded Undiagnosed Disease Network site at the University of Miami, and she will be reviewing the genetic causes of limb-girdle, how limb-girdle is inherited and passed on, and limb-girdle genetic testing strategies during today's discussion. So thanks for being here, Stephanie. Go ahead and share your presentation.
Okay, bear with me one second, everybody. Let me just make sure. Great. Can you see my screen, Nicole?
I can. Looks great. Thank you.
Okay, perfect. So thanks so much for the introduction, Nicole. So as Nicole said, my name is Stephanie, and I am a genetic counselor at the University of Miami. And today we'll be reviewing some of the genetics of limb-girdle muscular dystrophy. I've been attending this great conference so far, and so I know there's a lot of questions. So I'm going to try to answer some of the questions I've already seen, but hopefully we'll have time at the end to go over some of your questions as well. And so throughout the talk, we'll go over some genetics 101 to help set us up for the rest of the talk. And then as one of our previous speakers discussed, LGMD subtype naming. It has gotten a little confusing, so I'm here to kind of break it down for you. The inheritance patterns of limb-girdle muscular dystrophy, genetic testing strategies, and finally we'll end with discussing family planning and reproductive options. But first, I like to just go over what a genetic counselor is, because sometimes members of my family still don't even have quite a good idea of what I do. So genetic counselors, we have a master's degree in human genetics and or genetic counseling. It really just depends on which program you go to. And we're really trained to be a resource for both patients and physicians and other healthcare providers about genetic concepts. And I like to say we're kind of the link between physicians and patients. We're able to speak at all different types of levels. And we're involved in ordering genetic testing and disclosing results to these patients. And where some other providers don't really spend as much time as genetic counselors do, we really like to focus on the psychosocial or the emotional implications of some of these results in having a genetic diagnosis in general. And so as promised, we'll go over baseline genetics 101. We're going to go back to some high school biology here. And so some of you may know our body is made up of cells. We're made up of millions and millions of cells, and we have all different types of cells throughout our body. So for instance, we have hair cells, eye cells, muscle cells, which are really what we're talking about mostly today. And in each of our cells, there's something called a nucleus, which is like the brain of the cell. And it's the brain of the cell because it contains all of our genetic information, or our DNA. And in humans, our DNA is packaged into these little packets called chromosomes. And so chromosomes are just really, really densely packed DNA. And what a gene is a gene is just a segment of our DNA. And genes are made up of 4 letters, A, T, G, and C. Don't worry about the rest of the word. Just 4 letters in our DNA, and these are put in different patterns and different lengths over and over again throughout our DNA. And the importance of genes is that they form something called a protein. And now this is different than the proteins we eat in our food. Proteins in our body are basically the workers. They are the substances that go out and do things in our body. And one thing I just like to make as a basically a comparison, I like to think of our DNA like a giant library. So the nucleus is the library itself. Chromosomes you can think of like the bookshelves. Genes are the books on those bookshelves. And the proteins are what we learn from the books. And so when we discuss mutations, what most people out there think are something like X-Men or something going wrong in the water supply, that's not it at all, okay? So all a mutation really is a change in a point in our DNA. So remember before I just said our DNA is made up of 4 letters. So we basically know what each letter should be in a gene. And so a mutation, all that means is instead of having a T at the location, someone has a C, or instead of having a G, someone has an A, and vice versa, or maybe a letter's missing. That's all a mutation is. And what I like to tell my patients is that most mutations actually don't impact us at all. They don't do anything. They just make us who we are. And sometimes, though, a mutation occurs in a gene that just can't handle having that genetic change in it, which leads to a genetic condition. And these are not what we call nowadays pathogenic variants. So actually, we don't really use the term mutation in genetics anymore, but I tend to use it when talking to my patients because that's what everyone has heard of. And so I know one of the earlier speakers showed a picture of chromosomes, but I like to use the picture of chromosomes just to kind of illustrate some main genetic points. So remember before I mentioned that our chromosomes contain all of our genetic information. And so you can see chromosomes come in pairs. So we have chromosome 1, 2, 3, 4, all the way down to 22. And these are the same in both men and women. And then these last 2 chromosomes, these are called the sex chromosomes. So if you have 2 X chromosomes, you're a female. If you have an X chromosome and a Y chromosome, you're a male. And so I mentioned we have 2 copies of every pair of chromosomes. And this is because we get 1 of each pair from our mom and 1 of each pair from our dad. So for instance, this one might have come from mom, this one from dad, this one from dad, this one from mom, so on and so on. And this is important because remember I said our chromosomes are where our genes are. So this also means we have 2 copies of every gene in our body. And now when we're ready to have kids of our own, our chromosomes line up and only 1 of each pair goes into the egg, 1 of each pair goes into the sperm. And that way when egg and sperm meet, the baby has the proper number of chromosomes, 2 copies of each chromosome. And this is important because that means for every gene, again, with limb-girdle muscular dystrophy, we have 2 copies of every gene. And so I just like this illustration because now we know how beautiful genes and genetics can really be. And so to really go into the naming structure for limb-girdle muscular dystrophy, I just want to give a brief background. So the term limb-girdle muscular dystrophy wasn't really mentioned until 1954. And that's when these 2 physicians, Walton and Nattrass, said, hey, wait a second, we have a group of neuromuscular disorders that are different than Duchenne and different than SMA, and they put them into their own group called limb-girdle muscular dystrophy. And at the time, back in the 50s, they were quoted to say these are likely a heterogeneous group of disorders. Heterogeneous is just a fancy word meaning there's most likely many different genes that can cause limb-girdle muscular dystrophy. And it turns out they were right. And then in 1995, that's when we actually found out a few of the genes that cause limb-girdle muscular dystrophy. At the time, we knew of about 4 genes. And so then we were able to say, okay, in order to make this easier on ourselves, let's create a naming system. So back in the day, we used type 1 or type 2 to refer to the inheritance, 1 being dominant, 2 being recessive. And then we would assign a letter based on the gene discovery. What this means is that the first gene to be associated with limb-girdle was called A, second B, so on and so forth. But then in 2017, we kind of hit a problem, and that's when we reached LGMD type 2Z. So we ran out of letters, so what were we to do? And 2018 is when this new classification system came into effect, where instead of using the number first, we're using a letter first. So for inheritance pattern, we're using R for recessive, D for dominant, and then the order of discovery for the gene is a number. And sometimes we'll also say the affected protein. For the purposes of today's session, I'm mostly going to be using types 1 and 2 because that's what most of you in the audience have heard your entire life with this condition. And so some things of note with the naming. Again, type 1 or type D, this is autosomal dominant, and this makes up a small subset of cases, about 10% of limb-girdle muscular dystrophies. Type 2 or R, as we call it now, is autosomal recessive, which makes up about 80 to 85% of limb-girdle muscular dystrophy. Now, for those of you good at math, you might say, wait a second, this doesn't equal 100, and you would be correct. And that's because we still haven't accounted for, sometimes it's really hard looking at a family history to figure out what inheritance pattern this is, or maybe it's a different type of inheritance pattern that is going way beyond the scope for today's session. And so the name overall of whatever limb-girdle muscular dystrophy one has, it really just depends on what gene is causing it. So for example, this gene SGCG, back in the old days, but still what most people are probably calling it now, it was type limb-girdle muscular dystrophy type 2C. Nowadays though, it's actually been renamed to limb-girdle muscular dystrophy type R5. And one of the previous speakers alluded to this, but that's because some of the previously thought of genes that cause limb-girdle muscular dystrophy, we've now actually figured out, wait a second, that's actually not a limb-girdle muscular dystrophy, and they've been classified into their own system. Now, I'm not going to go too much into detail, but this is just a chart that shows the new nomenclature, the new naming system for the limb-girdle muscular dystrophies. And it's also showing the old naming system, the gene involved, and the name of the gene or the protein that gets made from that gene. And so at the time of this paper, which was 2017, we knew of 24 recessive conditions and 5 dominant conditions. But new genes are being discovered every year. But one thing I just like to harp on with all my patients, honestly, I don't worry too much about the type. I always just say, let's just know what gene you have involved, because that's going to help you the most in the end of the day. Just knowing, okay, this is the gene, the name may change over time, but this is my gene that's affected. And so that's the one I always make sure my patients are telling their neurologists or their other healthcare providers, this is my gene that's causing limb-girdle muscular dystrophy. And some quick facts or fun facts with limb-girdle muscular dystrophy. It's one of the most common muscular dystrophies out there. And it's, at this point in time, we know of over 30 genes that are currently associated with the condition. But as I said, we're discovering more every year. So now let's go into how we diagnose limb-girdle muscular dystrophy. So back in the day, it was a very, very hard procedure. So in order to diagnose someone just with flat out limb-girdle muscular dystrophy, the physician would have to obtain the history, perform a neurological evaluation, perform laboratory testing, electrodiagnostic studies like EMGs, which are no fun, muscle imaging, muscle biopsies for some people. And then maybe if you were in the 90s, early 2000s, you got a genetic test after all this. So this was quite an involved process. But nowadays, we do a neurological evaluation. At that first appointment, most of the time we're ordering our genetic test, hopefully getting an answer, sometimes not. And if we don't get an answer, that's when we might do some of those more invasive testing. And so let's go into how someone inherits limb-girdle muscular dystrophy. And we're going to start with type 1, which is the autosomal dominant form. Autosomal is just a fancy word for meaning that one of the genes, the gene that's involved, is on chromosomes 1 through 22, meaning that men and women are affected equally. With autosomal dominant conditions, we can see it inherited from an affected parent. And this is the condition that technically does not skip generations. And when a person has an autosomal dominant condition, every time they have a child, there's a 50% chance that this gets passed on. And so this picture I just pulled up, this is called a pedigree, just a fancy word for family history. And when you look at a pedigree, when you see squares, that means it's a man, circles are women. And when you see someone filled in this case, that means they're affected. When you see a line connecting a man and a woman this way, this means they have children together, and lines up and down mean that that's that person's child. So with an autosomal dominant pedigree, this is what we typically see. In this case, this man was affected. He had 3 children, 2 of them were affected. This affected individual had children who were also affected, and so on and so forth. But also see that not every child they have has the condition. And typically, when you do not inherit the autosomal dominant condition, you cannot pass that along to your child because you do not have the mutation to pass on. This is just an easier way to sometimes think of autosomal dominant inheritance. So in this case, this is a man who is affected with the condition. And remember, he has 2 copies of this chromosome. And one of his genes on this chromosome, he has a mutation. The other one, he doesn't. His partner in this case, she does not have any mutations in that gene. So whenever she passes on the gene, the child is inheriting a good copy. So we don't really need to worry about her. Then it comes down to the father. 50% of the time, he passes on the mutation, and 50% of the time, he does not pass on the mutation. So, and that's why we say anytime you have a child, it's a 50% chance. Now, one thing I like to note with my families is that genetics does not have a memory. So every child is a new event. So you can have a genetic condition and none of your children inherit it, or all of your children inherit it. And that's because each pregnancy is its own event. It's the same with having boys and girls, right? So it's a 50% chance you have a boy, 50% chance you have a girl. You have families out there that have 5 girls, 5 boys. It's this, or 2 and 2, 2 boys, 2 girls. So it really just depends. And so a question I'm often asked in clinic is, sometimes I have a patient sitting in front of me with an autosomal dominant form of limb-girdle. However, neither of their parents happen. So how does this work? And this is due to a phenomenon that's called de novo inheritance. De novo is just a fancy word meaning new in this person. And so basically what happens in these scenarios is that an individual is born with a mutation that neither of their parents have. And nothing that the parents did or didn't do caused this mutation. This is a natural phenomenon that we see in the human genome. Anytime a new child is conceived, that child has mutations that neither of their parents have. It's what helps the human species evolve. However, most of the time, like I said, those mutations actually don't do anything. They just make us who we are. But every once in a while, that mutation occurs in a gene that just cannot handle it. And that's when someone can have an autosomal dominant condition that neither of their parents have. When a couple has a child with a dominant condition that was inherited in a de novo manner, for their next child, I always say that there's always a chance that this can happen again. So there's always about a 1% chance. And that's due to something called germline mosaicism. We don't have to worry too much about that. But basically what it means is that either the eggs or the sperm in one of the parents has that mutation in it. And so that's why another child can have the condition. However, for their child, the one who has the autosomal dominant condition, anytime they have a child now, it's a 50-50 chance because the mutation has been introduced into their family line. And so let's go to the autosomal recessive form. So again, autosomal just means men and women are affected. But recessive means that you need to have a mutation in both copies of your gene. And so this means you have to inherit a mutation from mom and a mutation from dad. And this is when we can see a condition skipping generations, meaning the patient in front of me has no family history of limb-girdle muscular dystrophy. And that's to be expected because the chance of 2 people coming together and having a child while being a carrier for the same recessive condition is not very common, but it does happen, right? So whenever 2 carriers for the same limb-girdle muscular dystrophy have a child together, there is a 25% chance that the child will be affected. So every time there's a pregnancy, it's a 25% chance. And then if someone has a recessive condition, anytime they have a child, no matter what, their child will at least be a carrier for the condition, meaning they have 1 mutation. And that's because they only pass on a mutation. However, carriers are not affected with recessive conditions. And this is just to kind of illustrate what a family history might look like with a recessive condition. Again, most of the time when I see a patient, it's just the last generation that's filled in and no one else. But what this is kind of trying to show is that this father was affected with, or grandfather was affected with the same recessive condition. So her father was automatically a carrier. This individual, her siblings, had the same recessive condition, so she had a high chance of being a carrier. And so these 2 were carriers, had a child together, and that child had the condition. When it comes to recessive inheritance, this picture is much better at showing it. So again, mom and dad are both carriers, they do not have the condition. Anytime dad has a child, it's a 50% chance he passes on the gene without the mutation, or a 50% chance he passes on the gene with the mutation. Same for mom, 50% chance for the mutation or 50% chance for no mutation. So for every pregnancy, there is a 75% chance that the child will not inherit the condition and a 25% chance the child will inherit the condition. And again, carriers are not affected. And a question I'm often asked, especially in limb-girdle muscular dystrophy, is sometimes there's many family members who are affected, particularly in the same generation, so like siblings. And sometimes we see family members who are not as affected as each other or who have kind of different symptoms. And this is a very common thing we see in genetics, and it's due to something called variable expressivity. Variable expressivity applies to both dominant and recessive causes. And basically what this means is that even with the same mutation in the same family, people might have different ages of onset, different parts of their body involved, different speeds of progression of the condition, and different levels of severity. And we think that this is because even though we share DNA with our siblings or with other family members, we still have our own set of genetic information that makes us unique, that is somehow interacting with the genes that are involved, that are mutated in our case. And so what this means is that some people might have some modifying genes that are helping either make their disease more progress more severely, or some who keep their disease a little more mild. And we're still not that smart in genetics to have figured out these gene interactions yet, but that's going to be something within, I would say, the next 5 to 10 years that it's going to start being studied a lot more. So now let's talk about genetic testing. So my favorite question is, why is genetic testing important? As a genetic counselor, I'm going to be biased and say it's really important, but most of it is because we have specific forms of limb-girdle muscular dystrophy, and specific forms may have specific management guidelines to that gene that's affected. Also for inheritance pattern and family planning, so knowing, okay, this is my condition, this is the chance that my siblings might have it, my children might have it, etc. And as the second talk today went over, possible treatment in the future, particularly with gene therapy. In order to get gene therapy, we're going to need to know what gene is involved. And so genetic testing, basically what we do is we obtain a blood or saliva sample. You can technically use a muscle sample, but most labs don't do testing on muscle biopsies. And also, I'm sure you don't want to go through a muscle biopsy if you don't have to. So most of the time it's just blood or saliva. It gets sent to the lab, and the lab reads the genes. So remember, I said genes are like a book. So they're just reading the genes to see if there's any spelling changes or mutations that they're seeing. Then they're analyzing these mutations to see, okay, do we think this would cause disease or not? And then they'll issue a report back to the patient and the genetic counselor and the doctor that says, okay, these are the notable mutations we found. And this will include the gene, the location in the gene where the mutation is, and what letters have been changed, hasn't been seen in anyone else, etc. And so when I see a patient nowadays in a genetic session, especially with limb-girdle muscular dystrophy, we start off with something called a gene panel. A gene panel is a genetic test that's looking at a preset number of genes. It can be anything from like 1 or 2 genes to over 100 genes. With the limb-girdles, typically it's around a 50 to 80 gene panel. And this is because, as we've been discussing throughout the day, sometimes limb-girdle muscular dystrophy might actually be someone else in a patient. We might look at a patient and say, oh, we think this is limb-girdle, but their genetic result is actually showing us something different. And so with a panel, what a lot of these labs do is they kind of put together all of the genes that can cause like limb-girdle, Emery-Dreifuss, et cetera, to make sure we don't miss anything. The pros of doing a gene panel is we're getting better gene coverage. Because we're looking at a set number of genes, we're able to read those genes a lot more deeply. It's more cost effective, and it has a shorter turnaround time. Like we can get the results back in about a month. The cons is that no panel is going to include all of the genes that can cause limb-girdle. And that's because we're learning more about what genes cause the condition every day. And so these panels aren't updated every few months. They're updated maybe every year or so. So sometimes you might miss a gene that's affected in your patient because it's just not on the panel yet. And also with a panel, if we get a negative result, we might have to redo this panel again a few years down the line as we learn more in genetics. So when I see a patient in clinic and their panel comes back negative, the next step we do is something called whole exome sequencing. So we have over 20,000 genes in our body. And right now we know about like what 3,000 to 4,000 of those genes do. What whole exome sequencing does is it actually is looking at all 20,000 genes, but it's really only focusing on the genes, again, that we know to be associated with neuromuscular conditions. And so the pros of this is we're analyzing more genes. So we're able to pick up those more newly discovered genes that maybe only one group has found in like Tunisia. However, and what's also good about an exome is we can do a reanalysis of the exome in the future. Some labs do charge, some labs do not. So what we do is we wait, okay, so the exome comes back negative still, unfortunately. We say, okay, let's wait a year or two, and the lab will reanalyze it. So we get new information from the same data. Some of the cons is there's less specificity. So, and it's a higher cost and a longer turnaround time. We're looking at many more genes than a panel. So it's kind of casting a wider net. When we get a genetic test result back, there's 3 different types of results we can get. The first is positive, meaning a known pathogenic mutation, disease-causing mutation, either 1 or 2, depending on the inheritance pattern for that gene, is figured out on this test. Second is negative, meaning we don't find anything. This does not mean you do not have limb-girdle muscular dystrophy. What this just means is that either the gene is not yet known, or the mutation there is just so tricky, our technology can't pick it up yet. And the final type of result is what I'm sure many of you might have had with your own genetic testing, is something called a variant of uncertain significance, or a VUS. This is when we find a genetic change, but we don't have enough evidence to say this is disease-causing or not disease-causing. When we get VUSs, this is when we might say, okay, let's actually test some family members. For instance, if this is a dominant condition and we find a VUS in one of the dominant genes and there's an unaffected parent who has it, then we can say, okay, this is likely nothing. Or if we have a disease, like what looks to be a really suspicious mutation in a gene, and my patient's mother is affected and we find it in mom, we can say, okay, this is giving us some more evidence. We might also do some more invasive studies at that point as well, depending on the type of gene we found the mutation in. If someone comes back with a negative genetic test result, again, I always like to harp on this. This does not mean you do not have a genetic condition. We can always do research studies, like our second speaker today was discussing whole genome sequencing. This is typically only under a research basis nowadays. However, clinically, most labs are charging like 20 grand for it, and insurances are not covering these tests. Reanalysis as well, or retesting every 1 to 2 years as we're learning more and more about the genes that are causing limb-girdle muscular dystrophy. And so when we do these panels, we know that there's some genes that are more likely to be identified than others, and these tend to be the ones that were discovered first, right? Because again, more people are going to have mutations in those genes. And so what I like about this chart is it's kind of just showing you, this was from a study in 2017 that the MDA and the Jane group sponsored. And out of about 4,600 people, 1,300 were found to have a mutation. So it was about a 27% success rate back in the day. Again, this is from 2017. But notice how from Kaplan-3 to like FKRP, this is making up a good portion of the individuals who have limb-girdle muscular dystrophy. And then when you're getting down to some of these other genes, for instance, 2U, right? So U is much lower in the alphabet. We're only seeing 5 individuals who have it. And that's what we're figuring out today. The more newly discovered a gene is, the less people that we suspect to have that mutation or a mutation in that gene. And I know this picture is actually shown during the second talk as well. I don't want you to freak out looking at this, but what I like to use this is to show all the different areas where someone can have a mutation in this, our muscle cell complex in order to have limb-girdle muscular dystrophy. So this is the cell wall. Everything in here is the cell. Everything out here is outside of the cell. And our cells have these like little places to get information from other cells that we could have the mutation. This is the actual muscle fiber itself. So notice this green like Pac-Man looking thingy. This is the most common form of limb-girdle. This is the calpain 3 gene, protein technically. And so there's other components in this muscle fiber as well that we're still learning about all the time. And anytime we find a new component in the muscle fiber, that technically means that could be another gene that could lead to limb-girdle muscular dystrophy. And what's also important to note is that sometimes the same gene can cause different conditions. And we see this with a lot of the limb-girdle muscular dystrophies. And so when you get a result back, you might have a gene that says, oh, this can cause limb-girdle, Emery-Dreifuss, or myofibrillar myopathy. It might have a lot of things. And what I always tell my patients is that we're diagnosing you based on the physical symptoms you're showing us, along with what the gene is telling us. Sometimes we also might know, just based on where the mutation is in the gene or the type of mutation it is, what type of condition my patient in front of me has. For example, this gene TTN or Titin, it's one of the biggest genes in the body. If you plug it into this website called Omen, which is basically one of the genetic counseling Bibles, it comes up with all of these different conditions it can cause: cardiomyopathy, muscular dystrophy, limb-girdle, autosomal recessive, myofibrillar myopathy, celiac myopathy, tibial muscular dystrophy. So again, I have to use what's going on in front of my patient with this information about what this gene can be associated with in order to help properly diagnose our patients in front of me. And I just want to spend the last few minutes of my talk today going over some family planning options. And I always start this by saying, this is up to my patients and their partner. So you out there, I don't want you thinking I'm telling you to do any which way of starting your family. That is a very personal decision, and that is up to you and your family. However, what I do like to mention is that the chance of having a child with this condition is going to come down to the type of limb-girdle muscular dystrophy somebody has. So again, if they have one of the dominant forms, that means anytime they have a child, it is a 50% chance. If they have a recessive form, it's going to depend on if their partner is a carrier for a mutation in the same gene or not. This is very rare for a partner to be a carrier for the mutation, but it sometimes happens. So I always recommend the partner get tested. If they're found to be a carrier, anytime they have a child, it's actually up to a 50% chance. And anytime they don't have the mutation, I say it's very low, but I can never rule it out. So that's why we always recommend partner carrier testing with the recessive conditions. And so one other thing that is unfortunate is that we need to know what mutation a patient has in order to accurately predict the recurrence risk for them and to proceed with some of the genetic testing options I'm about to go over. So if we do not know the mutation or we haven't been able to find it yet, unfortunately, we kind of just have to look at the family history and say, this is the chance that you might have a child with it, but unfortunately, we're not going to be able to offer any additional testing. So some family planning options, again, you can always conceive naturally, not do any testing. That's totally fine. Some people conceive naturally and then they test the fetus during the pregnancy to see if the fetus has limb-girdle muscular dystrophy or not. And this is through using either a chorionic villus sampling or a CVS or an amniocentesis. So these are two procedures done. CVS is about 11 to 14 weeks of pregnancy, and the amnio is about 16 to 22 weeks of pregnancy. And what we do is it's basically taking a sample, not from the fetus, but around the fetus to test if that fetus has the mutations or not. There's also in vitro fertilization with pre-implantation genetic testing, which will be my last slide. And finally, things like adoption, sperm, egg donation, etc. Again, there's no right or wrong way to start your own family. So just to kind of end my talk today, IVF with pre-implantation genetic testing. So what this entails is going through the IVF procedure. And again, this can only be done if we know the mutations. Once we have the embryos from the IVF procedure, we let them grow for about 5 days. So they're about like 50 to 100 cells at this point. There's a biopsy, which is what this picture is shown here, where it takes out a few of the cells, and we do genetic testing on those cells. And then we see if that embryo has the mutations that would entail it to have limb-girdle muscular dystrophy. And only those without the mutations would be implanted into the mother. So the pros are this is a very successful procedure with about a 98% detection rate. Cons are that it can be very costly and most insurances do not cover it. And there's no guarantee that you'll end up with an embryo that you can implant. Also with the genetic testing with the IVF procedure, sometimes grandparents, brothers and sisters have to be involved in order to help make the test. And so finally, how can you or someone else be tested for limb-girdle muscular dystrophy? You can always find a genetic counselor in your area on the NSGC, or the National Society of Genetic Counselor website. Literally all you have to do is Google, find a genetic counselor, and it'll bring you to this website. You can pop in your zip code. You can also consult with your neurologist, genetic counselor, or geneticist near your center, and they can help point you in the right direction as well. And I just want to thank the MDA for having me here to speak with all of you today, and all of you for bearing with me and listening to me chat about genetics. And with that, I'll take any questions.
I love that little slide. That's cute.
Thanks. I'm really proud of this picture.
That's really cute. Okay, well, we do have some questions, so I want to get to those. So this person was diagnosed with 1A, which showed LMNA gene being affected, and they didn't see it on the new naming convention that you had mentioned. Is LMNA still a muscular dystrophy or another neuromuscular disease?
So it's been given its own term.
Okay.
So I actually just looked this up before my talk. Oh, actually, let me just go back to the slide that they were talking about, because I figured I was going to be getting a lot of questions about this. So LGMD 1A has been moved out of the naming system. So the LMNA gene, it's a really intense gene and can cause a variety of different symptoms or phenotypes. So it's actually been given its own term. And so you might be showing limb-girdle signs by having the LMNA gene. I would suggest talking with your neurologist more about it and seeing if there's any updated guidelines that you should be following based on that.
Okay, that's interesting. Okay. All right. So a couple other things. This person says, my husband had a noted autosomal dominant gene on his gene sequencing panel. We have a son and a daughter. Should one be more concerned about their child's inheritance than the other?
So if the father has a dominant condition, anytime he had a child, it's a 50% chance that the mutation got passed on. So technically what I would say right now is that both their children have a 50% chance of having the mutation if they're biologically his. Okay. Does that make sense?
Yeah. If it doesn't make sense, I'm going to ask that that person please type in a little bit more with their question. This person was diagnosed in 2018 with LGMD1C through genetic testing. They have 2 daughters in their 20s. Should they be tested?
So 1C, yes. So that is actually another, so the LMGD1C is another one that you'll notice is now kind of out of this limb-girdle naming system, because again, that's been put into its own cohort. But yes, as a dominant condition, each of her daughters will have a 50% chance of having it. So that's something they can consider for themselves. As hard as it is for parents, parents can't force their kids to get a genetic test. Yeah. So what I always recommend is having a frank discussion with your kids about, hey, this is my diagnosis. And then your children can always go see a genetic counselor on their own to learn more about it and learn the pros and cons of genetic testing.
Mm-hmm. And how progressive is type 2E? This person stopped walking at the age of 12 and both my parents are carriers.
So for the actual disease course, I don't know that off the top of my head. That's a great question for their neurologist. But what we do see is that recessive conditions tend to show up younger than dominant conditions. And that's because with recessive, again, you don't have any working copy of that gene. So that tends to show symptoms at a younger age compared to some of these dominant conditions where sometimes it's a little bit older. But again, it's going to depend on the exact disease. So unfortunately, I can't really speak to 2E precisely.
Okay, and I wanted to get back to this question that was actually presented in this morning. This person had genetic testing done several years ago. It came back limb-girdle, but not conclusive with any subtypes. Would you recommend them get retested again?
Yes.
Or okay?
Yeah, so I would recommend retesting. So back in 2018, they probably had a gene panel, which is that set number of genes. And if it's uncertain, what probably happened is either based on their symptoms, we're still categorizing them as a limb girdle, or maybe a variant of uncertain significance was identified. So it's always a good idea to check back in with either the lab that tested you or the genetic counselor who might have been involved in the process to see if maybe it's time to do either a new panel or maybe proceed to exome.
And not to throw a wrench in the plans, but what if they can't? What if they moved? You know, what if they can't contact that original provider?
Yeah, so I would suggest trying to contact to see if they can get an old copy of the original report. Genetic counselors, we kind of have our ways of getting information.
Okay.
So if we can get a copy of the report, we can pretty much figure out what's going on just looking at the report and then saying, okay, you know what, let's just redo the test with the same lab, a different lab, or let's proceed to exome.
Okay. This person was 65 and diagnosed at the age of 10 with limb-girdle. No family genetics was found. How are they able to find their subtype? They live in the Washington metro area.
Oh, Washington metro. So I'm assuming that's Washington, D.C. If so, there's tons, and even in Washington, like Seattle area, there's tons of genetic counselors all throughout the area. What I would suggest is going to the NSGC website and typing in your zip code, and a genetic counselor in your area should be able to pop up and they should be able to help you. Also speaking with your neurologist who's been following you, they might be able to recommend, like, if a genetic counselor works with them sometimes, or if a genetic counselor is in their area who they recommend seeing as well.
Okay, and I just put that in the chat as well so folks can see that. And so this person is saying that there are only 1 out of 7 that have been diagnosed with type 2D with the same mom and dad. She was diagnosed at 30, didn't run in her family. She's the first one. Would that be a de novo example?
So if it's type 2, that means it's recessive. So
okay.
2D would be the SGCI gene. And this is going back to like what I mentioned earlier about genetics not having a memory. So you would think given 7, technically, let's just round it up to 8 kids, that 2 of them should have the recessive condition, right? Because it's a 1 in 4 every pregnancy. However, each pregnancy is its own event. So it was a 25% time each time they had a child. And it might have just been that this one sister was the one who inherited the 2 mutations and none of the other siblings did. Or if some of her siblings are younger, maybe they just haven't reached the point of being symptomatic yet. So her siblings can consider genetic testing if they want to know if they're going to be at risk or not.
She was tested in 05. Should she get retested, or is her original test conclusive enough?
It's going to depend on the test. There's a lot of things that are going to come into this. So no, it's okay, because 05 is kind of a while ago.
Yeah, I just want to make sure I get the answer.
Yeah, so what I would say is I would recommend they see a genetic counselor who can kind of take a look at the test and look at the actual mutations that were found. And the genetic counselor can kind of get a sense of, okay, these are 2 really, really well-known conditions or mutations. This is definitely what's going on. We can stop here. Or they might say, you know what, this test is a little out of date. Let's keep going. But I would say most likely whatever was found in 05 should be okay. But I've seen cases where I've actually reversed diagnoses. So again, I would recommend a genetic counselor for this scenario.
Okay. This person says they have 2J. My mom's siblings as well. My grandmother and great uncle also had the same. Would I need to be retested, and what is the new classification?
So 2J is Titan, and Titan is quite the gene. So I would need actually a little more information about this one. That sounds like a dominant inheritance pattern.
Yeah.
So what it could be is that, So that's a dominant inheritance pattern. So I'm not sure if, because what sometimes happens is, particularly back when testing was done a few years ago, it might have been that it wasn't actually a diagnosis of 2J. It could have been that maybe there was a variant of uncertain significance or only one mutation found in 2J. So I would recommend talking to your genetic counselor again and seeing maybe there was a misunderstanding when you got this test result, when it was originally disclosed, and if maybe there's more testing we need to do in that case.
Okay, there are so many subsets.
Yes, so right now, so again, I looked on Omen this morning and I saw up to like 26. And then I don't think the gene that the doctor in the second talk mentioned is in Omen yet. So there's many genes with new being discovered all the time.
But yet they're
again,
all,
as we get that late, these are going to be really rare causes. These are going to be like one or two families in the entire world.
but yet they all still fall under limb-girdle umbrella.
Exactly, because it's probably a mutation going back to that pretty picture of the muscle I showed. It's probably a component of this structure somewhere that's also involved. So it's still messing with this system.
Oh, okay.
Yeah, so we're still that, and that's why basic biology research is still so important, because we're learning more about what is interacting with the muscle cells in order to figure out, okay, which genes do we need to look at next in some of our limb-girdle muscular dystrophy families.
Wow, it's really kind of overwhelming when you get into it.
Yeah.
Is it possible for dominant forms of limb-girdle to be digenic?
So digenic means that two genes are involved, just for everybody out there.
Okay.
As far as I know right now, we don't know of any digenic limb-girdle muscular dystrophy cases. Digenic isn't actually dominant or recessive. It's basically two different dominant conditions happening at the same time that are working together. So some people might have two different limb-girdle muscular dystrophies. With my job, I've seen crazy things happen. Or it could be what we're learning and what we're seeing with like more of the CMT or the Charcot-Marie-Tooth. We haven't really gotten into it too much with the limb-girdles yet. Is that sometimes we have one dominant mutation, and then we're finding out, oh, having this other mutation in like a recessive gene might be impacting it. But again, we're not that smart enough in genetics to have that fully sussed out yet.
Okay.
But that's to come.
Okay. And I just want to be cognizant of our time. I want to get to one more question here. This person has 1B as in boy.
Okay.
Is it Lamin or Layman? AC is missing.
1B, I thought, so 1B is also, so 1A, B, and C are the ones that are missing now. These are the ones that have been taken out. Bear with me one second. I wrote in my notes somewhere which B stood for. All right, this is what happens when you have a lot of slides and then they changed the nomenclature on you 2 years ago.
Sorry.
No, you're fine, you're fine. Where'd it go? Here it is, okay. So I'm sorry, before, if I said 1A was LMNA, I was wrong. 1A is MyoT, 1B is the LMNA gene.
Okay, so they have one. Okay, so they have 1B, LAMNAC is missing.
Yes.
No way to check with past generations. I have it and my son has it. Is it certain his daughter will have it?
No, so anytime your son has a child, it's a 50-50 chance.
All right.
So the daughter, her granddaughter has a 50% chance right now of having it.
Okay. Okay. Perfect. All right. And I'll just end with this here. This person was tested many years ago and all test results were normal. And they had a muscle biopsy, which showed just muscle atrophy, excuse me. The last testing was in 2010 or 2011. They were looking for the CAD9 gene, but they couldn't find it. She was diagnosed with limb-girdle, no number, and none of her parents have had muscular dystrophy. So can she just have regular limb-girdle with no subtype?
No, so every limb-girdle we believe has a subtype.
Okay.
And that's something I think I forgot to mention. So that testing 2010 is basically the 1800s when it comes to genetics. What I would recommend for this patient is redoing the test,
Okay.
especially because it sounds like they only did testing for LGMD2A.
Okay.
Because notice how I jumped right into panels. Back in the day, we maybe tested one gene at a time, just based off of, and most of the time it was CAP3 because that was our best shot. So what I would recommend for this patient is a new genetics consultation, because there are much better tests out there that are going to be able to get a better sense. But again, if the genetic test is negative, we say they have LGMD unclassified. But again, they still have limb-girdle. They most likely have a subtype. We just don't know that subtype yet.
Okay. Does 1C equal, or does 1C mean that the CAV3 is missing?
It's going to depend on the mutation, but typically what it means is that they have one mutation in the CAV3 gene that has a pathogenic mutation in it that's causing it not to work. Okay. Sometimes we can have deletions within the gene. Sometimes a gene can be missing, but it just depends on their specific mutation. But it just means one of their copies is not working properly.
Okay. All right. Well, we got a lot of questions that we tried to get through. Thank you very much, Stephanie, for being with us today. We definitely appreciate all your time.
Yeah, thank you so much for having me, and have a great weekend, everybody.
Thanks.